Hi, and this is Kuzera.
I'm Miss Pranke.
Today we're going to extract some DNA from strawberries.
Yes, we are.
Okay, so strawberries are the perfect fruit, or really any organism, to extract DNA from because they have so much DNA inside their cells. We have 56 chromosomes in this bad boy. They're considered polyploidy. Poly means
Many.
and ploidy is representing the number of chromosomes. So this has many chromosomes. There's 7 chromosomes per set in one cell, or 7 chromosomes per set, so that would be 8 sets, 56 chromosomes per cell in a strawberry. So we have lots and lots of cells here. Every time you eat a strawberry, you're eating DNA. Did you know that? All right, so let's extract some strawberries, DNA. If you guys have strawberries at home, and if you have isopropyl alcohol or rubbing alcohol can work.
Do it with us.
And then, yeah, and soapy water. You can do this with us.
So you want to start by putting your strawberry in a bag and you want to smash it for 2 minutes. So you want to be able to get it into a nice liquid substance. So Ms. Kuzera and I are going to take some time to smash up our strawberry.
Yeah, and one thing that I always like to do is I take the stem off. You don't have to, you can. I would suggest pushing all the air out of the bag
is a good
so that
idea.
way when you're smashing it, you don't bust a hole in it. And then you're going to want to just push it on a desk and you'll get a nice slurry. Now, if you have a blender at home, just do it in a blender.
Yes.
Okay, so just smash it up. You want it to be liquid, not too many chunks. And once you get a nice slurry, a liquid with lots of the juices, not too many chunks in there, you're going to be able to get the DNA out. Now, there's a little bit of a reading for the background of this lab. Go ahead and read through that. That way, you'll have a little bit of knowledge as to why we're smashing the strawberry. There's a reason for that. Strawberries are great to use because they're very easy to smash and make liquid, but we're trying to get access to the cells, increase the surface area of that strawberry so that all of the cells are going to be able to be exposed to the next step, which is going to be adding soapy water. This soapy water, this is going to be nonpolar stuff. And we know that like dissolves like. And what are cell membranes made out of? Do you remember? Cell membranes? They're made out of plasma membranes, phospholipids. Those are also going to be non-polar. So we're going to add some soapy water to our slurry. Really just a couple tablespoons at home. You can just do a splash like we did. Let's press out the air.
Mix it up.
Yep. And then you want to expose that soapy water to the strawberries cells. Now this is going to dissolve the cell membrane so that all that DNA is now going to be free floating in this solution. Okay, so now how do we get the DNA by itself? Well, to extract the DNA from this slurry, we're going to push it through a funnel that has a screen in it. If you don't have a screen with a funnel at home,
you can use cheesecloth, which can be found at any grocery store.
Or a t-shirt, an old t-shirt. Really, you can use anything
filtering.
that will filter, a colander if you have that at home, and then you can filter through the juices. So when you do that, you're going to want to put something underneath to catch the juice. I'm just going to put a test tube. You can use a glass at home, and the juice should be pretty thin. We don't want too many chunks in here. We might want to grab an extra cup if we have a beaker or a cup somewhere, because we really only need a little bit of this. We don't want to have too much in here. In the meantime, we're going to get a cup. That's perfect. We have a good amount of strawberry juice. If you look here, we have probably 2-3 tablespoons of strawberry juice. Ready? 1, 2, 3, switch. And then we're going to add the material that's going to be able to repel the DNA out of the strawberry juice. So that's where your alcohol comes into play, the isopropyl alcohol or rubbing alcohol. You'll notice that I have some ice in this bowl. The ice, you want your alcohol to be cold. So you're going to pour the alcohol in very slowly along the side of your glass. We have a pipette to do that. So we're extracting out some of that.
The reason that you want to pour it on the side is so you don't break that surface tension of the strawberry, so that way you still get your isopropyl alcohol layer on top.
So I'm going to bring this really close to the screen so you guys can see this, but I'm going to spin, there it is. I'm going to spin this a little bit, and you're going to be able to see the DNA, that spider web looking material that's coming up into the clear liquid, that's the DNA. Miss Pranke's grabbing a stir rod, a little wooden stir rod. If you have a coffee stirrer at home, anything that has some kind of friction on it to help grab onto the DNA. So we have some wood that's going to work. The little splinters of wood will do a good job extracting that out. So you just spin it kind of like a spaghetti spin, and then you'll be able to pull up.
You notice though that I'm not going into the red, I'm staying above the red line for your isopropyl alcohol. So you get this gooey little substance on your wooden stick.
So this stuff right here, if I could scrape it off, we didn't really get a whole lot out of here. I'm going to actually break that tension there.
Do we want some more isopropyl alcohol in there? Yeah. Here we go.
Let's try to get a little bit more. Oh, there it goes. So, but this stuff that looks like wet spider webs in the clear solution, that's all the DNA.
That's all your DNA.
This looks pretty gross. That's DNA right there. There we go. So there's some more. So you just need to do all of these steps pretty slowly. Now the questions are, what is the structure of DNA? Why would this look like this? Why does the DNA look kind of stringy in this nature? So after observing this demo, you can try this at home, and then there's some questions that I want you to think about while you guys are doing this lab. Thanks for watching. Have fun. And stop.
Johari.
Johari.